Just as amino acids can polymerize and make long chains that we call polypeptides, nucleotides can connect one after another to each other. When they do this, they can either make a long chain of DNA or they can make an RNA chain.
Phosphoric acids can form diesters also. Okay, we can actually attach two alcohol groups to a phosphoric acid because there's two places where the ester can be formed on that phosphate. If you look at this, you can see that there's phosphoric acid on the left and then two alcohol groups. I can bring an alcohol group in on one side and remove a water and attach the R group, and to the other side I can attach the R group. This makes a diester. This is essentially what happens when we start forming DNA or RNA, is that we take a sugar, make a phosphate ester, and then attach it to another sugar, so it's a phosphate diester. And this is the DNA backbone. It goes sugar, phosphate, sugar, phosphate, sugar, phosphate, and that is basically the backbone for a DNA strand. And then attached to that DNA strand of sugar, phosphate, are the bases. And you can see essentially in DNA what you have is a nucleotide in a phosphate ester with another nucleotide, which is in a phosphate ester with another nucleotide, and on and on. So we have our nucleotides joined together by phosphate groups. The RNA backbone is identical to the DNA backbone, except that instead of having deoxyribose, we have ribose. And in ribose, you have the nucleotides also. You have the bases attached to the ribose sugar. The difference between DNA and RNA is that in DNA, we have cytosine, thymine, adenine, and guanine bases, and in RNA we have cytosine, uracil, adenine, and guanine bases. They only differ by one base, and that's, two bases, thymine and uracil. Thymine is only found in DNA, and uracil is only found in RNA. Another way to describe DNA and RNA is as a polymer of nucleotides, where the phosphate group of one nucleotide attaches to the alcohol group on the ring of another one. And so we can describe them as polymers of nucleotides. The DNA and RNA backbone is assumed to be understood when describing DNA or RNA, and all we do is we give the order of bases based on the first letter of the bases. The order always starts at the end of the backbone that has a phosphate sticking out, and that phosphate is then attached to the number 5 carbon on the ribose, and it progresses. The order of the base is given, then progresses until you get to the end of the polymer that has just a sugar there, or it would be a nucleotide without a phosphate, then bonding it to the next nucleotide. So this short piece of RNA has a phosphate at the beginning. You see that at the top. And then we have uracil, a uracil base, or a uridine nucleoside, then a phosphate, and then we have a guanidine nucleoside. Okay, so these are being attached to each other, and you can see the beginning of the strand is considered where the phosphate is. The end of the strand is where the sugar is with no phosphate attached. We call the beginning at the phosphate, the 5' end, and we call the end at the sugar the 3' end. So here is a DNA strand, and it goes from the 5' end with the phosphate to the 3' end at the sugar, and the order of this has thymine, then guanidine, then cytosine, and then adenine. And so we would write describe this strand as TGCA. A lot of atoms are described, a lot of geometry is described by the simple TGCA. DNA occurs as a double strand, and RNA occurs as a single strand. They differ by this in addition to differing in that RNA has only uracil and DNA has only thiamine in it. The DNA strand is a double strand, and the two strands hydrogen bond to each other, so that a purine hydrogen bonds to a pyrimidine in the other strand, and a pyrimidine will hydrogen bond to a purine in the other strand. It is always that way, a purine to a pyrimidine and a pyrimidine to a purine. The strands run opposed to each other, meaning that you have one strand when they're hydrogen bound to each other and wrapping around each other. One strand, if you're at the bottom of one strand, it's the 5' end, and the bottom of the other strand across from it is the 3' end. And so if you run from the 5' end on one strand to the 3' end on the other strand, as you're running along parallel to it, you're going from the 3' end to the 5' end. This hydrogen bonding between the two strands is base specific. What we find is that in DNA, adenine only hydrogen bonds to thymine, and guanidine only hydrogen bonds to cytosine. It is very, very specific, and it is based on the distances between the strands, and it must be a purine with a pyrimidine because those give you a specific distance that is needed. This is a picture of adenine hydrogen bonding to thymine, and you can see there are two hydrogen bonds there. There's an amino group at the top of the adenine that is hydrogen bonding to the carbonyl in the thymine, and you can see the hydrogen in the attached to the nitrogen on the thiamine is hydrogen bonding to the nitrogen in the ring of adenine. So there are two hydrogen bonds there, and you can see it is a pyrimidine hydrogen bonding to a purine. Notice that the chains are written, one is upside down and one is right side up. That's because the chain on the left is going 5' to 3', and the chain on the right is going 3' to 5'. This is showing you cytosine to guanine hydrogen bonding. The cytosine is on the right and the guanine is on the left, and you can see that there are 3 hydrogen bonds here. The guanine carbonyl oxygen is hydrogen bonding to an amino group on the cytosine, and the hydrogen that is attached to the nitrogen in the guanine group is hydrogen bonding to a nitrogen in the cytosine group. And then the amino group on the guanine has a hydrogen that is hydrogen bonding to the carbonyl on the cytosine. Again, notice the DNA backbone is inverted in one chain. You can see that the chain on the left is going 5' to 3', and the chain on the right is going 3' to 5'. Here are 2 short DNA strands showing you how they pair together. Notice the top one goes 3' to 5', and the bottom one goes 5' to 3'. Also, notice that adenine always hydrogen bonds to thymine, and guanine always hydrogen bonds to cytosine. This gives us a reflection from one chain to the other, because if it's a thymine in one chain, it's an adenine in the other. If it's an adenine in one chain, it's a thymine in the other. And the same thing with guanine and cytidine. Base pairing works so that every strand can replicate itself because it makes its reflection. Notice that when we have growth of the chain in replication, it always goes 5' to 3'. Adenosine triphosphate has a similar structure to what we've been seeing, except it is a nucleotide with 3 phosphates on it instead of just 1. We also have ADP, which has only 2 phosphates on it. And you probably already know that this is how energy is transferred from one place to another in your cells, that ATP will be changed to ADP by losing a phosphate, and when that occurs, a tremendous amount of energy is released, and your cell functions will use that energy for metabolic processes. Adenosine monophosphate, it's similar to the nucleotide that we've been looking at, except the phosphate group has come around and formed a ring with the alcohol group in the number 3 carbon. This is called CAMP, and it is what's called a second messenger. It is present in the surface of cells along with CGMP, the guanidine similar compound, and what will happen is hormones will activate this in cell membranes so that as to trigger something inside a cell. So do the in-text problems as you read, and do the following end of chapter problems, and there will be an online quiz posted for you to do.